Environmental pollution has always been an issue, such as arsenic poisoning in drinking water caused by overmining, illegal discharge of industrial wastewater, and accidental oil spillage into the seas. Despite the amount of effort and money the government put in, the results are still in fact with little success. One problem we found was that there is a lack of real-time environment surveillance platform that is cost-efficient enough to cover a large radius of land of sea. We are a group of scientists and engineers, architects from Oxford and Cambridge. Today I present you our project named Spectre, which is a palm-sized miniaturized sensor platform that allows real-time environment surveillance and non-invasive GI tract health diagnosis.
We are surrounded by a huge variety of microbes, and some live in your garden, in your kitchen, and even in your body. So as a single-cell microorganism, these microbes can detect the environment to search for food and to run away from predators. So the key concept of whole-cell biosensor is to hijack the sensing mechanism of bacteria and use them to detect compound of interest. So in this project, we managed to showcase 3 different kinds of whole-cell biosensor, each to detect gut inflammation signals, genotoxic compounds, and oil spillage. We also managed to show that our sensor have high specificity, high sensitivity in which we can detect down to nanomolar concentration range, and fast response time in which the whole sensing duration takes about 20 to 30 minutes.
We designed a miniaturized imaging chamber and printed it from 3D printer. We modified the design many times, so the end result you can see is much smaller and lighter than the traditional plate reader. We selected a variety of material to against the mechanical impact from outside and ensure a biocontainment. Using optical fibers and X-shaped chamber to eliminate the influence from incident light, and also by adding the multiple LED with different wavelengths in case the different fluorescent protein is used.
To measure environmental signals with a traditional biosensor, a typical user would need to travel for several miles before they can take a measurement. In a lab, the fluorescence is measured with a huge plate reader, which is prohibitively expensive, fragile, and immobile, which means the biological components within the sample may have denatured over the transportation, and this represents one of the major bottlenecks in applying biosensor to environmental measurements. In contrast, Spectre allows users to take on-site measurements. Once the bacteria have produced fluorescent proteins in response to a chemical signal, we then measure that fluorescence with a tiny spectrometer chip over here. And for the prototype, an increased amount of GFP translates into an increased proportion of green light, which is then compared to a reference level to make sure the observation is significant and not just noise. Moreover, Spectre Plus records data wirelessly, and this comes in handy for future gastrointestinal applications, where a wireless design means the user can simply swallow the device and take real-time measurements within their body.
Through building Spectre and Spectre series, we certainly learned a lot, and I know there are many more difficulties lie ahead for us to solve. In the future, we hope we can use the Spectre technology to change how pollutants being detected in the environment, and thus save our only one motherland. If you're interested in us, please stop by our booth and ask for more information. Thank you.